How's it going? Good morning. Good morning. Nice to see those of you who could make it this morning. Looks like about half the class. Okay, let's see. First of all, Dr. Lavasani got called out of the country like the day before she was to lecture you guys. And so that's never happened before since I've been here. But the material that she would have covered is on last year's ECHO, and you did get her slides. And so you're still going to have to be responsible for that material for our exam, for our final. Now, Dr. Lavasani is expected to be back on Monday. So if you've gone through the material by then, you might want to contact her if you have any questions. That was kind of unfortunate. On your exam, the average after we made the adjustments was 82.86. For all the systems courses, our exams, for like the entire sophomore year, or D2 year, that's about where the averages are. They're usually between 82 and 85. And that's probably where they should be, somewhere in the middle of the B range. And you guys all got into dental school, so that means you're really good students. So the average should be at least, in the Bs or higher. So all in all, you did, pretty much on this exam as other classes have. For those of you who received a grade less than 70, nobody's grade is to the point where you're in serious trouble. You just have to work harder so that you can get it over that hump. So I would encourage you to do that. The final involves more material. There's more faculty involved. But I can say that for most of the lectures, and definitely for my lectures, there's no outside material again. It's all going to be covered here in class. If with a grade change, like with the last grade change on your exam, if your grade did not go up, it's because you were already given credit for that. Like if we say everyone gets credit, well, if we'd already given you credit, then then your grade wouldn't have changed. Are there any questions about any of that? Yes?
When you're making an exam,
yes.
How do you pick the topics that you want to make questions over, and then could you just talk about that?
Sure. We divide up the exam according to lecture hours. So if I say, on this last exam, I think it was approximately 8 questions per lecture hour, something like that. We try to make the exam come out to be about 50 or 60 questions. And then we divide up the number of lecturers by the number of hours, and we distribute it that way. It doesn't work out perfectly. Some of the material in some of the hours we need to cover a little bit more, and some a little bit less. And one thing that really surprised me, though, is I specifically pointed to 2 formulas and said, you're going to have to pick out this formula for hydroxyapatite, and 2 of you got both of those formulas wrong. So I'll try to underline this is on the test, so that's a little closer. But that really surprised me. But anyway, it happens. Anyway, did that answer your question? Does that answer your question? So sometimes you ask me, are there images on the exam? Well, I'm not putting any images on the exam, but some of my colleagues may at the last minute decide they want to. Yes? Pardon? Yes. It's just It's just the way it is. It is. It is, and I've been discussing that with one of the students in the class. I think for next year, I'm going to try to see if I can get another week or two added to the course without adding more material and see if we can get one more exam in there. It falls at this unfortunate time of the year in a way. It's the end of the year. There's only so many weeks left. The good thing is, right now, some of your classes have ended already, so you may have a little more time to focus on this. Yeah, I understand what, I hear you. What you're saying is true. It's a lot of material, and but it is what it is. For the most part, on my material, I try to not make the questions so detailed that you have to memorize all these lectures. That obviously wouldn't be possible. So for today's, are there any other questions? So today we're going to be talking about muscle. And some of what we're going to be talking about is so basic you probably heard about it, and you probably talked about it in grade school. But there will be a few things that you haven't, and hopefully it will be useful to you. But to start with, the most basic things are the functions of muscle. It helps determine body shape, provide voluntary movement, involuntary functions. Temperature regulation and heat production is not something often that people often think of when they think of muscle, but it's very much involved in keeping your body temperature regulated, and then obviously protection. So body shape, obviously, muscle can be a big determinant of body shape, from this guy here down to this selfie I took of myself. No. I'm probably closer to that guy. Voluntary action, obviously you have some control of your muscle action, as this person does. Maintains you in an upright position, allows you to stand and walk, et cetera, et cetera. There are involuntary muscle actions. Your heart beats, whether you like it or not, and presumably you like it. Digestion, breathing, reflex actions to heat, et cetera. All of these things are involuntary actions of muscle. And temperature regulation. Muscle produces heat, and it assists in keeping your body temperature stable. If you're standing on a corner and it's very cold and you're waiting for a bus, you may find yourself hopping around. This is because every time a muscle contracts, it gives off a certain amount of heat. So shivering, for example, or movement on your part, you know that when you're running, you're warmer than when you're standing still. So muscle, we'll go into this a little bit more at the end of the day. And protection. Protect the body in emergencies. It surrounds the visceral organs, but also your muscles can protect you by helping you fight off dangers. I want to be with this guy. Do that. All right. Muscle can be classified in different ways. Now, you all know that there's cardiac, skeletal, and smooth muscle. This is their general structure. We'll talk about this in more detail. But the classification of muscle can depend on whether you're talking about its function or its structure. Now, in terms of function, there's voluntary and involuntary muscles. And you'd consider in the involuntary muscles the cardiac muscle and the smooth muscle, because your heart beats on its own, and smooth muscles such as in digestion, for digestion, there is smooth muscle around all of the blood vessels in the body with the exception of capillaries. And they are intimately involved in the regulation of blood flow to various tissues and back to the heart. And so these are involuntary functions. And then, of course, there's the voluntary functions of skeletal muscle. So muscle can be classified this way functionally. But if you're talking about structure, sometimes you'll see it classified as striated or non-striated muscles. This is basically, based on these striations or bands that you see in this skeletal muscle at the top and this cardiac muscle in the middle. And then these type of banding is not seen then in smooth muscle. So if you're talking about structure, striated and non-striated may be a way that they're divided up. So it really depends what you're talking about. And so, again, these are the three types of muscle tissue, and cardiac, skeletal, smooth, and cardiac. And we're going to start by talking about the skeletal muscle. Okay, location. Where's skeletal muscle? Oh, attached to bones. Yeah, sure. Okay, so long, cylindrical, multinucleated, and it's heavily striated. It's voluntary, and it produces movement at joints, stimulated by nerves, etc. It controls the movement of the skeleton. It's under conscious control, and it makes up about 40% of the body weight. So we're going to, you've had this in anatomy, but we're going to go through a little bit of the structure of skeletal muscle. Around the outside of a muscle is the epimysium. It's this layer that surrounds this this bundle, if you will, of muscle fascicles. So each one of these is called a fascicle. Around them is the epimysium, and each fascicle is surrounded then by the perimysium. So this little blue band here is the perimysium around each fascicle. This is a fascicle, and then this is the epimysium. Then within each fascicle are bundles of muscle fibers, and these are surrounded by these what's called the endomysium. So there's endomysium, perimysium, and epimysium. And there's fascicles and fibers. Okay. So this shows that same diagram here. What we see then is there is also surrounding each of these is a sarcoplasmic reticulum. Sarcoplasmic reticulum is like a sac, very much like the endoplasmic reticulum within cells, only it surrounds these muscle bundles, and it's responsible then for releasing calcium into the area of the muscle filaments in order for them to contract. So we'll show in detail how this all works. So here is, there are thin and thick muscle filaments. The thin filaments are called actin filaments, basically, and the thick filaments are called myosin filaments. But in fact, the actin filament is made up of several different proteins. In this picture here, the red spheres are considered the actin molecules, and they're kind of strung together in this helical structure on this more filamentous tropomyosin. So the tropomyosin is holding these actin molecules together, and periodically along this tropomyosin there are troponin complexes. They're shown in yellow here. Now these troponin complexes then are responsible for changing the conformational arrangement of this actin, tropin, tropomyosin complex. So when calcium binds to troponin, it kind of twists this molecule in such a way that it exposes binding sites on the active molecule that can interact with myosin and allow the muscle to contract. So that's one of the things that allows muscle contraction to occur is this binding then of calcium to troponin, specifically to troponin C, this unraveling it a little bit and allowing then this myosin head here on the thick filament, this myosin head then to bind or to attach to this actin molecule in order for it to contract. And then the striations then in the muscle are basically these related then to the regions where there's either overlap of actin and myosin, or there's just actin or just myosin. So here is what's called the Z line, which is kind of where the actin filaments are anchored. There's a zone where there's, we would see a band of basically just actin, which would be like right here. Then there's a zone where there's an overlap of the actin and myosin. Then in the middle, where there's no overlap, the actin kind of ends here. This would be just a band of myosin, and it's bisected by this so-called M line. And so you see how these striations then are where they come from. This just shows what I was talking about in a little bit more detail. Here's our actin shown in green. Here's our tropomyosin filament that forms this kind of helix. This whole thing's called the thin filament. And here's the troponin that's going to be responsible then for exposing these sites on actin by twisting it so that the actin can bind then with this myosin head here. That just shows the same thing. Troponin is not a single molecule, it's a complex. It's the troponin C that binds calcium, however. So let's look at this, how this works then.
With calcium elevated around the myofibrils, myosin binding sites on actin are exposed, allowing interaction of the myosin cross bridges with actin. Release of the myosin cross bridge from actin requires binding of ATP. After ATP binds, it is split to ADP and inorganic phosphate. The energy derived from this reaction is utilized to caulk the cross bridge in preparation for the power stroke. The power stroke is initiated when the myosin cross bridge binds to actin. The cycle begins anew when another ATP binds to myosin. At rest, most cross bridges are in the caulked configuration, prepared to interact with actin when the tropomyosin troponin complex is moved away from myosin binding sites on actin by calcium ions.
Okay, so the calcium ions bind here like this. Then ATP comes in and allows and supplies the energy for the actual contraction. Okay. No question about that? Straightforward, right? Probably seen that before. Okay, so what we're going to talk about now is excitation-contraction coupling. That is, how does a stimulation from a nerve or a motor neuron then cause the muscle to contract? And we're going to start at the muscle and kind of work backwards. So here in this part of the diagram are thick and thin filaments. Okay, so here's the blue, then is the thick filament of myosin. The pink is the thin filament of actin, which contains the troponin and tropomyosin. And then you see this whole thing this whole fiber is surrounded by this sarcoplasmic reticulum shown here as this blue structure here. An action potential is generated along the surface of the muscle, and by that we mean that nerve stimulates the surface of the muscle and it depolarizes. That is, there's an influx of there's an exchange of sodium and potassium along the surface, which depolarizes it. And this depolarization is carried all along the surface of this muscle fiber and then into these regions called T-tubules. These are just like invaginations of the fiber around the outside of the muscle, and they invaginate into the T-tubules. And so it conducts then this action potential inside the muscle, or into an area very close then to the sarcoplasmic reticulum. So you see there's a very close relationship then between the membrane on the outside of the muscle and the sarcoplasmic reticulum. So this is how the action potential is communicated then into the muscle. What happens then is that the sarcoplasmic reticulum is stimulated to release calcium. Calcium then floods across these fibers, and it causes that reaction that we just saw in the video, where the calcium then, binds to troponin C, exposes a site, and allows this allows this interaction to happen. So this shows it in more detail. This is the surface membrane here. There is a sodium, when the membrane depolarizes, sodium rushes in and potassium rushes out. To restore the membrane for the next contraction, there is a sodium-potassium pump that pumps the sodium back out and the potassium back in so that this cycle can be repeated. But after this, so we see this action potential then travel across the surface of the membrane into the T tubule until it gets to a voltage-sensitive calcium receptor. The voltage-sensitive calcium receptor, called the DHP receptor, or an L-type calcium channel, allows calcium then to enter into the muscle cell, but it's kind of a relatively small amount of calcium. Its main function then is to trigger this ryanodine receptor here. The ryanodine receptor then is kind of like a door that opens and allows massive amounts of calcium to flood out of the sarcoplasmic reticulum onto the myofilaments in order for contraction to occur. So this is like the triggering event for the ryanodine receptor, which allows this large amounts of calcium to flood in. There are a couple of places where ATP is required for muscle contraction. One is, as we saw in the video, is for the myosin head to actually move. So that requires ATP. And also to pump this calcium back into the sarcoplasmic reticulum after contraction has occurred. So both of these things require energy, or ATP. This here is actually an exchanger, and I don't believe this requires ATP. So how do we depolarize this surface membrane in the first place? Again, we're moving backwards. Well, here's the membrane, here's the T tubule, and the way it becomes depolarized first is at this neuromuscular junction. Okay, this is an axon terminal here, and this is the neuromuscular junction, and so this is where there's the connection then between the nervous system and the surface of the muscle. This shows that same area in more detail. All right, here's this myelinated axon motor neuron. When an action potential is generated down this neuron and it gets to this neuromuscular junction, there are acetylcholine molecules that are in these packages here that are along the surface, along the terminus of the axon. When this membrane depolarizes, these acetylcholine molecules are released into this cleft region between the axon terminus and the surface of the muscle. When this happens then, it causes then, this is called the motor end plate, by the way. When acetylcholine is released into this area, receptors open that allow sodium to flood in and for the action potential to start. So here's our action potential coming down the nerve. It gets to the nerve, the axon terminus. Here, these red dots are the acetylcholine being released into this cleft at the motor end plate. These are channels that are opened then in response to acetylcholine, and then they then allow sodium in and potassium out and depolarize this membrane to move it down the chain. This is an actual image of what they look like. All right, so let's see this all together.
The process of excitation-contraction coupling consists of all those processes involved between the time of generation of the action potential on the muscle sarcolemma and the actual contraction. As the action potential reaches the neuromuscular junction, vesicles containing acetylcholine fuse with the membrane of the nerve terminal, and acetylcholine is released into the cleft. Acetylcholine molecules rapidly diffuse across the cleft and bind to the acetylcholine receptors. Binding of acetylcholine to receptors immediately opens ion channels, allowing sodium ions to rush into the muscle fiber and potassium ions to diffuse out. Acetylcholine is then split to acetate and choline by cholinesterase, resulting in inactivation and closure of the channel. The initial flux of ions due to opening of the channels brings the membrane potential to threshold, causing the voltage-gated sodium channels in the vicinity to open. The resulting action potential is propagated over the entire surface of the muscle and also courses into the T-tubule system. As the action potential encounters DHP receptors in the T-tubules, it causes opening of the ryanodine calcium channels of the lateral sacs, thus allowing calcium ions to diffuse out of the lateral sacs and into the region of the myofibrils. Calcium then binds to troponin, resulting in movement of the tropomyosin-troponin complex away from myosin binding sites on actin. The contraction process continues as long as action potentials continue to reach the neuromuscular junction, thereby keeping the concentration of calcium elevated around the myofibrils. To end the contraction, calcium must be actively transferred by the calcium pump, calcium ATPase, back into the lumen of the lateral sacs. This pump utilizes ATP as a source of energy. This process reduces the concentration of calcium in the vicinity of the myofibrils, such that calcium dissociates from troponin. Tropomyosin again covers myosin-binding sites on actin, and the muscle relaxes.
Pretty straightforward, right?
Actually,
Yes.
so what's the purpose of the sodium flooding in?
That depolarizes the membranes and it propagates the, it changes, the membranes are polarized, you know, they're positive on one side and negative on the other. And by depolarizing them, then it propagates that action potential along the surface. It allows the entire muscle to contract at one time.
And what was the receptor of the T-tubule? DHP?
DHP receptor, that's an L-type calcium channel. That's that allows calcium to come in and stimulate the ryanodine receptor on the sarcoplasmic reticulum to release large amounts of calcium. Okay? Okay? All right. Okay, this structure is called the muscle spindle. This is the receptor. This is a receptor for stretch. This is a stretch receptor in muscle. There are extrafusal fibers, which is this outside part here. That's basically normal, regular old muscle. The extrafusal fiber is basically muscle, but in the spindle, so the spindle itself is surrounded by these extrafusal fibers, which are basically normal muscle. The intrafusal fibers are those which is what is unique about the muscle spindle. These fibers, in this case, they're wrapped with this type 1A afferent, which basically detects the stretch of the muscle. So if you pick up something like this, you know, I can pick up this piece of paper and I can pick up this cup, and the action of my arm is identical, but the action of my muscles is not, because the cup is heavier than the paper. So there has to be an adjustment then in the response, and that's what occurs at the muscle spindle. So by detecting the stretch of these interfusal fibers, then there is an adjustment made in the amount of contraction that occurs. So this kind of gives a bit of an idea of how this works. These represent action potentials, or this is the firing of the nerves. And so as you stretch the muscle, then there's more nerve stimulation that occurs from these sensory fibers that then that goes to the brain, and then that sends down a signal to contract then the extrafusal fibers, or the rest of the muscle, in response to the amount of stretch. So the more it's stretched, the more you resist the stretch by in response. So for example, if you're holding onto your child's hand in the mall and you're walking along, and if he's walking right next to you, your muscles are kind of relaxed. But if he tries to take off, your arm becomes that your muscles contract, your arm stays in the same position, and the kid can't go anywhere because you have contracted muscles in response to the stretch. As you feel the stretch, you're resisting it. So the muscle spindle responds that way. There are multiple types of fibers that serve various functions here, but you know, for this class, you've got enough to worry about without, but just you need to know, understand though, that this is what the muscle spindle does. It's basically the stretch receptor, okay? There is also another type of stretch receptor called the Golgi tendon organ, and it's, as you see, it's located in tendons. This has actually an inhibitory effect on muscle stretch. What this does is it keeps you from ripping your muscle away from your bone. So the Golgi tendon organ does not like to be stretched. When it feels tension at all, it sends inhibitory impulses to the muscle to tell it to relax so that it doesn't rip itself off the bone. So, so that these two are kind of opposite in that regard. Any questions about that? So the spindle responds by increasing stretch in response to load, and the Golgi tendon organ kind of inhibits stretch in response to load. Okay. The muscle tone is basically, muscles stay in a state of partial contraction kind of all the time. If you cut the nerve to a muscle, you know, sometimes you'll see somebody, you know, after a stroke or something, their face will droop or whatever. That's from the loss of muscle tone. They're no longer in the slightly contracted state. In other words, the resting state of a muscle is not, you know, without any signal at all. So this is physical exercise that improves tone of muscle and increases their size. You know, we can see that. Those of us, not me, but those of you who are in shape, you see the muscle tone. You know, the muscles remain in a nice type shape. And then those who do not exercise, their muscle tone decreases, and they don't look as good, I guess. So an ideal muscle loses its tone and wastes away. If a person does not use certain muscles or use them very little, the muscles become flabby and weak and may atrophy and waste away. This, I believe, is what happens in space. This is why I want a trip to Mars. This is something that must also be considered. I talked about how bone will, if a person's in space for a long period of time, they will lose bone and it will not come back. They will also lose muscle. If you have, you know, here on Earth we have gravity pulling on us all the time. We have our own weight, you know, pushing on our muscles and our bones, and this then helps to develop tone. If you are in an environment where where this is not necessary, those muscles will atrophy and can eventually waste away. So this is another thing that must be considered for anyone who's you know, going to survive in an environment where there's no loading of their muscles and their bones. This describes two different types of muscle contraction: isotonic contraction and isometric contraction. Isotonic means like the same tone. So muscles, if you lift a weight, as in this picture, then the weight doesn't change. So that from the time you pick it up and flex your muscle, the weight itself isn't changing. So the load isn't changing. What's changing is that as the muscle contracts, the muscle is shortening and thickening, but the load is not changed. In an isometric contraction, such as, you know, pushing against the wall, you know, I can push harder and harder. I hear the wall cracking, so I don't want to do that. In this case, the wall isn't moving, and you know, I'm not even moving, but the muscle is contracting. The harder I push on the wall, the more contracted the muscle becomes, the more muscle tension. So, but there is no shortening. So in this case, this is supposed to represent a table or some object that isn't going to move. And so as he pulls on this muscle is contracting more and more by this isometric contraction, but there is no movement occurring no shortening of the muscle itself. And in this case, there is shortening of the muscle, but the load is constant. Okay, is that clear? Okay. Now there are different types of muscle fibers. There's slow twitch and fast twitch, and these have been subdivided into, there's, in modern literature, there's many different types of fibers, but for our discussion, I just want you to understand the distinction between a slow twitch type muscle fiber and a fast twitch type muscle fiber. Slow twitch muscle fibers stain very dark, and they're smaller than fast twitch muscle fibers. The reason that they are is because they contain more mitochondria. That is, they are better for long-term type of endurance. So here, the slow twitch fiber then, for things that require endurance and prolonged strength, if you have a muscle that's predominantly slow twitch fibers, you will see that there's more capillaries associated with it. More aerobic activity are in those type of muscles. They have more myoglobin, which is kind of like hemoglobin. It's a molecule that can kind of store oxygen in muscle. And more mitochondria. Remember, mitochondria is the source of ATP for muscle contraction and for everything else. And so an example of this is the soleus muscle. You remember the soleus muscle at kind of the base of the calf? That allows you to just stand for great periods of time, lecturing to sleepy students, and without falling over. So this particular muscle is predominantly made up of slow twitch type fibers. They have this endurance and prolonged strength, and you can go for longer periods of time. Now, the fast twitch type fibers, which are the lighter ones here, they're bigger, first of all. They can be almost twice as big as the slow twitch. And these are more in muscles for power, for maximum power, for things that are kind of activities such as jumping or kicking or something like that requires a lot of power but for a short period of time. The gastrocnemius muscle, like when you're, which again is a little higher up on the calf, for jumping, that's a muscle that has a lot of type 2 fibers. Okay, the type 2A fiber is both aerobic and anaerobic, and the type B fiber is strictly anaerobic. And so, do you understand kind of the difference between those, right? Okay. So this shows something about contraction speed. We see that the type 1 fibers for endurance, their contraction is much slower. This is milliseconds. It's 200 milliseconds from the time it starts to contract to it ends, in this case. This is the gastrocnemius muscle. This is mostly type 2, and it has maximum power, but a much shorter time frame. And this is another muscle, extraocular muscles in the eyes. These move extremely rapidly and change extremely rapidly. So it gives you an idea that, I guess I'm belaboring the point, but that's what we're talking about here. Now this, I think, is actually very interesting, and I think you might find this interesting too. Back in 50s or 60s, when they could do this, they could never do this experiment today, but they could do it then. In some university, I'm not sure which one, they took a bunch of athletes and they biopsied their muscles. These are students who, wanted a good grade. And so when they did that, they took all of these different types and they biopsied them, and then they did this kind of differential staining to look for fast twitch and slow twitch fibers. Remember, this has got a lot of fast twitch, or the darker fibers. This is a lot of slow twitch fibers, and this is somewhere in between. And what they found is, if you notice, for the marathoners who are predominantly involved in endurance type activities, they had an abundance of slow twitch muscle fibers when they were sampled, and fewer fast twitch. For those athletes that were sprinters or jumpers, they had this reversed. All right, so there was a higher amount of fast twitch fibers and a higher amount of less amount of slow twitch fibers. Here's our average guy here, so almost half and half. Okay, well, what makes this interesting, though, is that there's very little switching of fiber types, almost none, in a person's muscles. What that means is that if you're born with a large percentage of fast twitch fibers and a smaller percentage of slow twitch fibers, the exercising and all doesn't change that. So why I think this is interesting is these marathoners with a high amount of slow twitch fibers and less amounts of fast twitch fibers, they selected themselves to be marathoners. They didn't decide that they would, run and then they would develop all these slow twitch muscles and then they would become marathoners. What happened was when they were in school and they had field day, they found that they had more endurance than other people and they could run longer and they didn't mind it. So they self-selected to be marathoners because they had more slow twitch fibers. They didn't develop them because they liked running marathons. The same with the jumpers and the sprinters. The sprinters found that, wow, I can really outrun that guy who just lasted longer than me, but I can run, short periods much faster than him. So that's why I find this interesting. So these athletes were self-selecting based on their genetic endowment. But I suppose that's not all that unusual. I mean, Michael Phelps chose swimming because he had really long arms and really big feet. Yes?
So I know you can't gain like the amount of fiber that you have, but can you train the ones that you do have to perform better? So like athletes, if they do have, a less amount of fast twitch fibers, can they train them to perform better?
Well, I mean, sure. When you exercise, you build muscle. And you improve muscle tone, and just obviously exercise improves your performance. But what this is saying is that they're starting at a different, at a higher level. That what they may be able to achieve in their life may be limited in part by the genetics of this relationship. That being said, there is under certain conditions where you can redistribute this a little bit. Any other questions? Okay. Now this molecule I find fascinating. This molecule is called myostatin. Have you guys talked about this at all? Myostatin is one of the coolest molecules ever, and I'll show you why. It's also called GDF8, growth something factor 8. I don't remember what the D is. It's produced in muscle cells, and it reacts with muscle cells. I think the line at the bottom isn't on your slide. I put that on for clarification. So in other words, myostatin is produced by muscle, and it binds to receptors on other muscles, or on muscle itself. And what myostatin does is it inhibits muscle growth. But I want to digress 1 minute because I think this is really funny, and I think you might find this interesting. In molecular, in MCBM or everywhere else, they talked about SMADs. Did you ever remember hearing about SMADs? Yes? No? Well, the only reason I bring it up is because the derivation of the name SMAD is really funny. SMAD was it's a molecule in a pathway, and it was discovered. The MAD part of SMAD was discovered in Drosophila, those fruit flies. Well, it turns out that fruit flies have 15 appendages, 15 antennas and legs and wings, and they're 15 altogether, okay? When they would do mutations in the SMAD pathway, those appendages wouldn't work. So when this was discovered, they called this SMAD because the MAD stands for Mothers Against Decapentaplegia. That's what MAD and SMAD stands for. Decapentaplegia means, having 15 appendages, none of which work. So Mothers Against Decapentaplegia is what that stands for. I just thought that was pretty funny, but I don't get out much. Okay. Anyway, so back to myostatin. So myostatin binds to its receptor and inhibits muscle growth. So what do you think would happen if you had a myostatin, if you took a mouse and you were to inhibit myostatin? What do you think the mouse would look like?
Double muscles.
Double muscles. Okay, well, here's a normal mouse muscle with the skin stripped away, and here's a myostatin knockout. Here's a normal mouse muscle. Here's a myostatin knockout muscle. Here's the normal mouse, and here's the myostatin knockout. So when you look at that, the next thing you wonder is, well, could this happen in other organisms? What would happen if this happened? Well, it turns out that there are both natural and genetically altered organisms where they have knocked out myostatin, and this is what you get. This is called a Belgian blue cattle. And yes, that's all muscle. This dog here, this is a greyhound. That's a greyhound, one of the skinniest dogs there are, and that's a trout. Okay? So this is, now, in the case of the Belgian blue, this was a natural, this occurred naturally. I mean, it wasn't done on purpose. I'm not sure about the dog, but the fish was done on purpose to try to get more meat on the fish. The problem with this is, I mean, even though, you know, you think, wow, that'd be pretty good for steak, the problem is it causes, there's other health problems associated with these. These animals are kind of fragile. And so there's reason why myostatin is suppressed. So then the question comes up, well, what about a human being? What if, is there anyone who has had a spontaneous mutation in myostatin? Well, this baby here was born with a gene mutation without myostatin. And you see this little muscle on this baby's a little bit larger. Okay? When this kid grew up a little bit, this is what it looks like. This is what happened to him. So now I know, yeah. So this seems like the ideal bodybuilding thing. But in fact, there are medical problems associated with this. But yeah, this child has a natural myostatin mutation that it caused him to build excess muscle mass. I don't know how old is he here. He's grown up now, but it's pretty incredible, you have to admit. Okay, we're going to stop here for right now. Take about a 5-minute break, and then we'll continue.
Because it's like a genetic kind of thing, or do they just weaken over time?
No, because bone responds to load by increasing too. So with this added load from the muscle, the bones also increase to accommodate. Yeah, the problems are less concerned with the skeleton, with like other hormones and other organ systems, the circulatory system. Well, they don't adapt quite equivalent to handle this. Sure.
Regarding the lecture. So for this slide, I don't really understand the difference between when a muscle's contracted, relaxed, and stretched. Could you just explain that one more time?
Okay, this is the impulses, the nerve impulses. Mm-hmm. Okay, so what this is saying is when this muscle fiber, which is the receptor, mm-hmm. is being stretched, mm-hmm. it's sending a huge amount of impulses up to the brain, and it's responding then by contracting the surrounding muscle to kind of resist the load. Okay, so. So if you try to lift up something really heavy, it's sending a lot of signals to cause the muscle around it to be able to deal with that really heavy thing. Okay.
So muscles around it, as in the extrafusal?
As in the extrafusal and the rest of the muscles.
Okay.
The extrafusal is is basically normal muscle around the spindle, but then all the other muscle too.
Okay.
It's like a spring, right?
Yeah.
So when it's stretched, it's telling the brain, hey, tell the extrafusal to bring me back.
So you go to lift up something that's really heavy, and as you start to lift it, more and more action potentials are going until the point where you can actually get it off the ground. But so it's sending signals back to say we're not going to be able to lift this unless we have more and more and more and more and more.
Okay.
Okay.
That's helpful. And then my next question. Could you explain the difference between these two again? So isotonic shortens and thickens the muscle, so it causes.
Okay, so I'm listening to this by isotonic. This muscle's contracting. This, it's iso means same, and same load. This muscle has the same load, which is the weight of this all the way through. But so the muscle's shortening. Isotonic, same load. Okay. Isometric is if I will try to lift this desk. The harder I push, the more my muscle contracts, but the desk isn't going anywhere. So isometric means that the length of the muscle isn't changing. The metric or length of the muscle isn't changing. So it's not shortening, but it's increasing the tension. Yeah, the tension increasing. Yeah.
So we're not considering the angle of the weight, like it's actually lighter at the top.
Well, this is just the basic definition for these two things to distinguish the two. You know.
And then my last one was you talked about the gastrocnemius muscle, which helps in jumping. Is that type 2A or B for fast?
It's kind of both.
Okay.
Dr. Borke,
Sure.
where'd you say the Z line was? It's just actin?
Oh, it's just where the actin is anchored. You know these are just a landmark. I'm not sure what it's made out of, but the actin are anchored at the Z line. So if this is the Z line and this is the Z line, the actin are anchored like that, and then the myosin is between them. And then when they contract, they pull the two Z lines together, basically, okay?
And the M line is where the myosin is?
Right down the center, yeah, on the center, the myosins are anchored kind of down this Z line in between, so.
Sorry, we know you need a break.
No, I wouldn't, that's fine.
It's okay if I
Yeah,
guess?
sure.
More? Okay, so when we have the action potential in the muscle fiber, it binds to DH2 of the T-tubule. Then something happens with calcium, and then we have a power stroke. So what happens with the calcium?
Okay, so here's the membrane here, and we have sodium potassium going across, and it's changing the polarity. Say it was positive on the outside and negative on the inside, it's switching so that it's electrical-like, transferring down there. Gets to the DHP receptor, which is this calcium channel. That lets a calcium molecule from the outside in, and that is the signal to open the ryanodine receptor. And the ryanodine receptor is then like a door that allows the flooding of a boatload of calcium out of the sarcoplasmic reticulum over the muscle fibers for the contraction. In other words, it's not the calcium from the outside as much that's causing the muscle contraction. That's just like a signal to signal the internal calcium. And so there's two receptors involved. The DHP receptor is the receptor on the muscle surface, and the ryanodine receptor is the receptor on the sarcoplasmic reticulum, and they work together.
Okay, so the ryanodine receptor, so it goes into the T-tubule.
I can show you that slide again.
Yeah, that was that's new information. I don't know.
Okay. Let's see. All right. So here's the T-tubule. This is the DHP, or voltage-dependent calcium. So this is the calcium from the outside. Here's the outside of the cell, and the calcium goes in, and it triggers the ryanodine receptor to release all of this calcium to cause muscle contraction.
Okay.
So this receptor interacts with this receptor.
Gotcha.
This is the two receptors.
Gotcha. That makes a lot of sense. That's all. Thank you.
All right. Sure. All right, I guess we'll get started again, if you would not mind. Okay, what I'm going to talk about next then is smooth muscle. We talked about skeletal muscle. Now we're going to talk about smooth muscle. Some of the characteristics of smooth muscle are similar to those of skeletal muscle, but some are very different. Smooth muscle is located on the walls of hollow organs, vessels, respiratory passageways. Again, the entire cardiovascular system, all of the blood vessels in the cardiovascular system are surrounded with smooth muscle with the exception of capillaries. A smooth muscle cell is tapered at each end and has a single nucleus. Okay, remember a muscle fiber could be quite long, but smooth muscle cells are individual cells with single nuclei. Primarily, they're involved in involuntary movement, and they produce this, kind of peristalsis that occurs in intestines, helps maintain blood pressure, etc. They are involved in the internal organs of the body, movements involved with that, propelling urine through the urinary tract, food in the digestive tract, dilating the pupils of the eyes, contracting blood vessels, etc. Remember, so this is a smooth muscle cell, one nucleus, okay? There's no tropomyosin, there's no troponin. But there are what are called dense bodies that are analogous to the Z line, that is, and I'll show you that in a minute. And they have a type of ATPase. Remember, for the myosin head to move, it required ATP. And there is there's an actin and myosin interaction that occurs in smooth muscle, and it requires this ATPase. And there's also very little sarcoplasmic reticulum. Yes?
Did you say no tropomyosin?
Yes, well, significantly less. So let me show you how this works. This is a smooth muscle cell, and the filaments then, the thick filaments are here is these, shown here as these kind of dense bodies, and then the thin filaments are these blue, all right? So during contraction, then again, the myosin and actin slide across each other, but what they cause a contraction more like this, okay? They're kind of, squishing the cell if you want to see it that way. And so this is a very different kind of muscle contraction, but it still involves the thick and thin filaments. So it is still a muscle contraction. So excitation-contraction coupling then is very different too. Calcium ions from the outside of the cell play a bigger role in the contraction of these. Calcium from the outside, they bind then to an enzyme complex called the calmodulin myosin light chain kinase. That's this here. So here's calcium coming in from the outside, and this membrane becomes depolarized. Calcium comes in, and it binds to this calcium calmodulin myosin light chain kinase complex. That's what this purple and green thing is here. And this enzyme basically breaks ATP into ADP and inorganic phosphase directly. So this then is what causes the contraction, okay? So here we have an unphosphorylated myosin light chain. This ATPase then allows then this to become phosphorylated, and then this myosin ATPase, this active myosin ATPase, will cause this cross-bridge cycling and the contraction to occur. So myosin forms the cross bridges with actin, as in skeletal muscle. When the calcium is pumped out of the cell, the phosphate gets removed from the myosin by another enzyme, and myosin becomes inactive and the muscle relaxes. So it's kind of a different process. We see there's also calcium coming from in this case, from the endoplasmic reticulum. Endoplasmic reticulum and sarcoplasmic reticulum are basically two names for very similar structures. So when calcium enters, then calcium from the outside and from the endoplasmic reticulum bind to this complex and basically phosphorylate the myosin light chain, which allows it then this to occur. Now there's two different kinds of smooth muscle, what are called multi-unit smooth muscles and single-unit smooth muscles. The muscle cells look similar, but in the case of the single-unit smooth muscle cells, and it's easy to confuse these two because the single unit contains a bunch of cells all together. So you might think that this would be called the multi-unit, but it's not. It's called a single-unit smooth muscle because there are gap junctions that connect the smooth muscle cells and allow it to contract as a single unit. Okay? So this is called a single unit because it contracts together as a single unit. The multi-unit, there's individual smooth muscle cells, and each of these cells contracts individually. Now, the way that they're stimulated is the autonomic nervous system. Then these are nerves. You see that there are these varicosities or these widenings all along these nerves. They don't have like a neuromuscular junction as a single structure on the end of an axon. Instead, they have these bumps all along their surface. And so in the multi-unit smooth muscle, there's these nerves with their varicosities kind of weave in and out between these individual muscle cells. And what this allows then is for very fine control and gradual responses. So in other words, if there's a single action potential comes down here and only a couple of these varicosities release their acetylcholine or into the area of the smooth muscle cells, then only a few of these cells will contract. So therefore, you have a lot of control, but depending on how much signal is coming in depends on how many of these are going to contract. So you have a lot more nerves and you have this fine motor control. In the case of this single unit smooth muscle, you see there's really only one of these, or they're kind of along the surface of this large group of muscles that are all connected by gap junctions. And so when the transmitter is released and binds to this outermost layer of cells, that change in action potential on the surface of these cells is conducted to all the other cells through the gap junctions. The gap junctions are a way then for communication to occur between these individual cells. So you don't need to have these varicosities in association with the surface of each of these cells, only with the outermost ones, and then it will transfer that signal to the inside. So that acts as a syncytium. A syncytium is when a group of cells act as a single cell, or a group of something acts as a single entity. And so that's what's happening here. So this is the difference between single unit and multi-unit and single unit smooth muscles. So this just kind of reiterates, in the multi-unit smooth muscle, in this organization, the smooth muscle cells are mostly separate, richly enriched with neurons, and they're found in places where fine control of contraction is needed, such as respiration and in arteries. The single unit smooth muscle is organized. The cells are connected by gap junctions. There's fewer neurons, and this is where larger areas need to respond all together, such as in the gastrointestinal tract and in the uterus. That's all I'm going to say about smooth muscle. Anything. We do cover more of this when we talk about the cardiovascular system. And then cardiac muscle. Cardiac muscle is obviously located in the walls of the heart. It has branching networks of fibers with a single unit. There'll be a single nucleus for a series of branch fibers. It's also under involuntary control, and its role is obvious. It pumps blood out of the heart. It can be self-excitatory. In other words, cardiac muscle can react, an action potential can be generated on the surface of cardiac muscle spontaneously, but it is also highly influenced by the nervous system and by hormones. Now, there are different layers to the heart. There's the epicardium on the outside, and there's the endocardium on the inside. There's even a pericardium, which is a sac that goes over the epicardium. But the myocardium refers to the muscle layer. So cardiac muscle is the myocardium, which is the muscle, obviously. Again, it's syncytial in that the fibers are branched and interconnected. So here's a nucleus of a cardiac muscle fiber. You can see that there's branching occurs between these different fibers. The fibers are the individual cardiac muscle cells or fibers are separated from each other by these structures here, which contain multiple gap junctions. These are called intercalated discs. So this structure here, this is an intercalated disc, which is what these blue lines are in this poor picture here. These then are covered with gap junctions, and they have a large surface area because they've got these finger-like interdigitations and a lot of gap junctions. And this then allows them to communicate with each other, just as we saw in the single unit smooth muscle, to allow then propagation from cell to cell. Gap junctions can communicate both electrical signals and can communicate ionic exchange across these between cells. Here we see this branching and the single nucleus here, but the branching. And mitochondria are kind of located between the fibers. Excitation contraction coupling has some differences from skeletal muscle excitation contraction coupling. For one thing, it's stimulated by a pacemaker potential. In other words, the heart pumps kind of spontaneously. That is, there is a node in the heart, in the right atria of the heart, that produces a pacemaking signal, or an action potential that then is propagated through the heart muscles. It's propagated along a sarcoplasmic reticulum. It goes down through a T-tubule, just like it does in skeletal muscle. There are calcium channels that open then and allow calcium in and stimulate a ryanodine receptor, just like in skeletal muscle, which then can allow calcium from the endoplasmic reticulum or sarcoplasmic reticulum then to release calcium to flood across the myofibers, just as in the skeletal muscle. One of the things, though, that's different is there's a much more reliance on the extracellular calcium. In other words, a lot more calcium in cardiac muscle comes from outside and is responsible for the stimulation and the contraction than it is in skeletal muscle. In skeletal muscle, the majority comes from the sarcoplasmic reticulum, and the calcium channel on the skeletal muscle is kind of just like for signaling. But in this case, the calcium coming from the outside is very important in this contraction. Also, so these are the three main differences that are important. Stimulated by a pacemaker, it's more reliant on the extracellular calcium, and also the action potential is much longer. For skeletal muscle, it's about 200 to 400 milliseconds, whereas it's in cardiac muscle, it's 200 to 400 milliseconds versus 2 to 5 milliseconds for skeletal muscle. And this is due to a mechanism whereby there is a prolonged influx of calcium. In other words, when the action potential is populated or propagated along a skeletal muscle, the calcium floods immediately, the muscle contracts, and then the calcium goes out. That's not how it works in cardiac muscle. There is a retention of calcium for a while. The calcium lingers, if you will, which makes this action potential longer. And this shows what I'm talking about here. The blue is a skeletal muscle action potential in about 2 milliseconds. Okay? They both respond then to this immediately, both the skeletal and cardiac, to this change in action potential along their surface. But then the skeletal muscle goes right back to and could be stimulated again, whereas the cardiac muscle does not. It has this prolonged period, this prolonged lag period, 500 milliseconds, which then prevents a second action potential from being propagated immediately. And that's important because of this phenomenon here, which is there's three concepts here: summation, trepe, and tetanus. Basically, for a skeletal muscle, if you attached it to an electrical stimulus and you started cranking up the frequency of the stimulus, so you start here. By the way, a contraction of a muscle cell is called a twitch. That's the physical contraction. So each of these represents a stimulation, and muscle twitch occurs. That's how it normally looks. If these stimulations become closer together, you get this what's called trepe. And trepe means basically step. And you see that these action potentials kind of get closer and closer together, and they basically summate or build on each other. This is what we mean by summation. They're starting to increase. If these action potentials get even closer, then it gets to the point where you see that the twitch does not go back down to baseline. They're piling up on each other, basically. So if we look back at this slide, basically the second action potential would be occurring here, and the third here, and the fourth there. And so what happens then is you get these contractions building on each other to the point where if there's fast enough stimulation, the muscle stays completely contracted, and this is what's called tetanus. So this happens in skeletal muscle, but this does not happen in cardiac muscle. And the reason it doesn't happen is because of this lag period in the action potential. You cannot get a second stimulation of a cardiac muscle because of this delay in the action potential. And this is very important because nobody would want their heart to go into tetanus if they just were overstimulating it. So this kind of shows that there is a refractory period that occurs, and this is a period where even if you had more stimulation of the heart muscle, it would not contract. It's refractory to contraction no matter how many stimuli are in here. This is what's called the relative refractory period. It means that there can be a premature contraction occur at this point if the signal's high enough. And that's what we see here is this premature contraction occurring here. Okay? So force of ventricular heart muscle contraction showing also the duration of the refractory period and the relative refractory period. So that's the refractory period, no second stimulation can occur, and relative is it can, but probably the contraction won't be as great, and it'll be somewhat abnormal. Okay. So how does this long action potential work? Well, the initial phase here, called phase 0, is when the sodium depolarizes the membrane. And phase 1 is when the potassium goes out. They occur right on top of each other. But what's unusual then in cardiac muscle is there's this phase 2 where calcium enters the cell to initiate the contraction, and then it takes a while for the calcium pumps to pump that calcium back out and back into the ER, and this causes this delay in repolarization, so that the complete repolarization, or this return of potassium back to the cell, occurs much later, before it goes back to the resting potential and can tolerate a second action potential. This is the electrical activity. This is the contraction. So this is membrane potential in millivolts. And we see this refractory period. This is muscle contraction. See, it's in tension. This is the tension of the muscle. And we see that this then is responsible for this refractory period. And one of the reasons this is so important is because of the way the heart must contract. The heart, blood returns to the heart then through this inferior and superior vena cava into the right atrium of the heart. This SA node here is the pacemaker of the heart, and it then sends a signal that is transferred across the right and left atria of the heart, which cause it all to contract like a syncytium. See, I put the word atrial syncytium here, so they contract together. But the whole heart doesn't contract, only the right and left atrium. There is this kind of insulating layer of fibrous structure that does not allow this action potential to move from the atria into the ventricle directly. There is instead a single passage for this action potential to go from the atria to the ventricle, and that's through this structure here. There's this SA node is the pacemaker that causes the initial contraction of the right and left atria. The AV node then, as this action potential goes across the atria, it eventually stimulates the AV node, which then transfers that action potential through this, what's called a bundle of His, down through these fibers, these Purkinje fibers, to the left and right ventricle of the heart. And so this is the only connection then between these two, the atria and the ventricles. What this does is it causes then a delay in the contraction of the ventricles. This is how this delay occurs. So blood comes into the heart, the right and left atria contract, they force the blood into the right and left ventricle, then the right and left ventricle contract. So this allows those two contractions to be separated in time. Okay, and so the bottom line is that this is the main difference between cardiac muscle, a functional difference between cardiac muscle and skeletal muscle, is in this delay that that allows there to be a timely contraction of the muscle. Are there any questions about this? Yes. Abnormal heartbeats, they can be due to, and we go into a lot of this in the cardiovascular system. There can be all of the above. There can be a problem with the either the pacemaker itself can be messed up, the AV node can be not receiving signals as much. But there can be what's called a right and left bundle block. That's when a right bundle block would block this nerve here. A left bundle block blocks this nerve. There can be, if you have myocardial infarction, or you can have a portion of dead muscle here that can affect it. There can be valve problems that can cause differences. So basically, this is a mechanical and electrical structure, and anything that mechanically and electrically can go wrong can. And a lot of what a cardiologist does is tries to determine where is the problem. Is it the valve? Is it this electrical conduction? Is it the muscle? Is it whatever? So what you're asking is a key point, and we do go into that in the cardiovascular system. Okay. Okay, so, all right, the contraction of all three types of muscle require energy. And where does the energy come from? Well, the energy comes from food. And so food is basically fuel. It comes through the intestinal tract and into the blood, and then it goes into the muscle cell, and it's converted to energy in the mitochondria. So this chart over here basically shows glucose or glycogen, carbohydrates, fatty acids, amino acids, and any other substrates. They are basically converted into acetyl-CoA to form ATP. And ATP then is used for all of these things here: muscle contraction, secretion, synthesis and growth, nerve conduction, active absorption, transport of materials across membranes. All of this is important, and basically it all comes from food through the action of mitochondria, which I'm sure you studied somewhere else in the curriculum. The immediate source of energy for muscle is onboard ATP. That is, ATP that's in the muscle, ready to go, is the immediate source of ATP. So when it becomes hydrolyzed It then releases its high-energy phosphate bond, then is used then for the contraction of actin and myosin filaments. That's the immediate source of energy. The problem is there's almost, it's not really a problem, but the way this works is there's almost no free ATP in muscle. So this immediate source is maybe good for one contraction, but it's very, there's very little onboard ATP ready for contraction. So, and you need ATP for two things, basically. You need ATP then to return the calcium back into the sarcoplasmic reticulum after the contraction, and you need ATP then to cause the myosin to move against the actin. So for that, the myosin ATPase in order for the contraction to occur. So you need ATP in two places. The immediate source is onboard ATP, and there isn't much of it. So there is, fortunately, what's called, you could call a short-term reserve of ATP. And this exists in the form of this phosphocreatine, or creatine phosphate. That's just a different name for the same thing. And what at rest, then, in the mitochondria, there's an enzyme called creatine phosphokinase. And creatine phosphokinase in membrane, in the mitochondria at rest, takes ATP and takes the high-energy phosphate bond and puts it onto this creatine molecule to form this phosphocreatine, or creatine phosphate. So this high-energy phosphate bond then is short-term stored in this phosphocreatine. So there's more of this available. And to use this for energy, then you simply cleave this high-energy phosphate bond off of here, and then it can be used then for energy. So there are two isoforms of the creatine phosphokinase. There's this one that is in the mitochondria, which at rest then causes the formation of the phosphocreatine kinase. And then during exercise, this is the enzyme that's in the myosin head that's responsible then for cleaving that high-energy phosphate bond off the ATP. So during exercise then, this ATP is now available for this creatine phosphokinase isoform that's to reverse this process and make that high-energy phosphate bond available for the contraction, for the movement of that myosin head. Okay. Creatinine is like a breakdown product. When muscle is sick, sometimes they'll be able to detect creatinine in higher levels than normal. Creatinine, just as an aside, is a useful molecule in studying kidneys, in that creatinine is neither secreted nor resorbed by the tubules in a kidney. So if you were to inject creatinine into the bloodstream and then measure creatinine in the urine, you can measure glomerular filtration that way, because it won't be absorbed and it won't be secreted by the kidney. So if you're wondering where you've heard this before, we'll talk about this in the renal system, but this can be used for measuring renal glomerular filtration through the kidney. But anyway, that's an aside. This is the part that we're concerned with here, where we're making the short-term ATP reserve by storing the high-energy phosphate bond on the phosphocreatine during rest and then taking it off during exercise. At rest, the primary source of the ATP is actually fatty acids. Fatty acids don't make a lot of ATP. I mean, they make like one molecule at a time. It's kind of slow. But at rest, they're the primary way that source where the ATP comes from. And I'll talk about that a little bit more in a minute. We talked about immediate source, then this is a short-term source, and then the sustaining source for aerobic exercise, aerobic source of energy is the Krebs cycle. And this occurs in the mitochondria. And then there's anaerobic source of energy is glycolysis, and you should have studied these. Basically, these are sources of ATP and energy for contraction. And this then is kind of like a summary. This is our immediate ATP going to ADP and releasing this much energy. Here's our phosphocreatine plus ADP going to form creatine and ATP. So this is, this is our short-term source. Here's our anaerobic source from glycolysis. From this, you see here we get 4 ATP from glycolysis by anaerobic respiration. Here we get these 40 ATP from the Krebs cycle through anaerobic respiration. And here's the formation of a single ATP by the oxidation of free fatty acids. As I mentioned, this is where the ATP comes from at rest. So these are basically the sources of ATP. And the last thing we're going to talk about today, I guess, is heat generation from muscle. Most of the body's heat is generated from muscle activity. When you're very active, they can draw on the stored glycogen in their cells. The muscle cells store large amounts of glycogen, which is basically a storage product of glucose. Glucose, when you're not too active, and if you take in more glucose than you're expending, it will go to fill up your glycogen stores in various organs in your body, of which the liver is a major one. And so when you become cold, then it takes the ability of this muscle to produce heat by both voluntary and involuntary muscle contraction. Involuntary would be shivering. Voluntary would be hopping around, for example, or running or slapping yourself, okay? Something like that. Any muscle contraction can generate some heat. So that's what we mean by heat can be produced in this way. Now, infants, however, do not shiver. They do not generally shiver. So they have a special mechanism for the heat generation, and it's from what's called brown fat. This is also true of small animals such as squirrels and the like. The little creatures then, they maintain their body temperature not necessarily, in part through this brown fat mechanism. And this is the location of brown fat in an infant. Okay, and how does it work? Over here is a normal white fat. This is normal fat in how it normally works. Okay, so say adrenaline. Your cold, adrenaline or epinephrine binds to your cells here. It goes then and it converts these free fatty acids. They go to the mitochondria, and there is this coupling that occurs between hydrogen ion movement into the mitochondria and the production of ATP. So this then, as a result, produces ATP, produces energy, and then we as an adult will take this energy and then maybe we'll use it to jump around or to shiver or whatever, just to try, to keep ourselves warm. This is a brown fat cell on this side. When adrenaline binds to this cell, the free fatty acid goes into the mitochondria. There's an active protein called uncoupling protein. This UCP stands for uncoupling protein. And it uncouples the formation of ATP from this breakdown of the free fatty acids. And this uncoupling of this then causes the release of heat. And so the presence of this uncoupling protein then is the source of the heat in the brown fat. Okay, so small animals and small people then, when they become cold, rather than the energy being used for you to motivate yourself to, either throw a blanket on, run into a store, whatever. Since the babies can't do that, they just generate heat by this through this uncoupling protein. Okay, are there any questions? That's all I have for today. Yes?
At what age do we make the shift from using brown fat to white fat?
The exact age, I'm not certain, but it's around the time that a baby starts to be able to crawl, and so we do have some brown fat also, but its function in us is not known. But the importance of it ceases to be important once the child starts to move around on his own. Are there any other questions? Okay, Dr. Bohm will be in here at 10, so you have a little time. All right, you all have a good day. Okay.